a lot of things are going to be similar between the first lab and this lab. Obviously, we're dealing with chemical compounds, okay? So all the safety stuff that we did in the first one still applies. You got to wear your safety goggles, you got to have proper footwear for tomorrow, okay? All of that kind of stuff. But what we're investigating about these chemical compounds this time is different. Last time, we were just looking at their physical properties. This time, we're mixing them with something else. That's a chemical properties thing. Okay? We're trying to see what happens when this thing is around this other thing, okay, and what is going to get produced. That's essentially what we're investigating here. So the problem is when substances are mixed, the chemical properties of each material determine whether or not a reaction will occur. Okay? What evidence do we have that a reaction takes place when substances are mixed? We talked about the signs of a chemical reaction last Wednesday. Right? When we talked about chemical reactions, we said, what are the signs of a chemical change? And we went over those. You guys know what they are. It's a review from junior high. Okay? So what we want to look at here are, what are manipulated responding controlled variables? Remember this time to do this. Okay? That is to explain your choices. No one needs to lose 3 marks needlessly this time. Okay. Each time, but essentially the procedure is going to work like this. There are pairs of things you are going to have to mix together. Okay? So you can see that down here in the procedure. Okay? Pair 1, you're going to mix A and B. That would be sodium hydroxide and ammonium chloride. Okay? You're going to mix those 2 things together. And then in pair number 2, you're going to mix C and D. All right? So that'll be acetic acid with sodium hydrogen carbonate. In other words, vinegar and baking soda. Yeah, okay, you've done that one a few times before. Okay, A and E will be your 3rd pair, so that'll be sodium hydroxide and magnesium chloride, okay, and so on and so on. So we've got these different pairs that we're going to be mixing. Each time we mix them, we're going to be watching for what happens. That is, we're going to be looking for the signs of a chemical change. Okay, things like color change, odor change, bubbling and fizzing, temperature change. Okay, is a precipitate being formed? So there's a change in solubility. All of that kind of stuff. Those are the signs of a chemical change that we are looking for. If we see any of them, we know that a chemical change occurred. Everybody follow there. Okay. Each time we do one of these pairs, what should we make sure is the same each time? Yeah, we should probably mix equal amounts. Okay, and we're going to do a pretty good job of that because we're going to have scoops and we're going to have eyedroppers. And you're only going to be able to take so much in a scoop or an eyedropper. So it's going to make pretty sure that you're using about the same amount every time. Now, in our first lab, one of the problems was we were dealing with incredibly small amounts of everything. Okay, we're rectifying that for this lab. You're going to be dealing with bigger amounts of everything, and you're only going to be mixing them one at a time. It's not like they'll have to be in that tiny little spot plate. Okay, and you'll be mixing them in there because that would be really inefficient. So what you're going to get is what's called a watch glass. It looks like a big contact lens. Okay, it's about this big around. Okay, and it's like a dish. Okay, but it looks like a big concave contact lens. Right. So it's going to sit on the counter, and you're going to do the mixing in that particular watch glass. So, manipulated variable, that's the thing we're changing. Each time, it's going to be a different what? A different pair of materials, I would think. Okay? Each time we do this experiment, it's going to be with a different pair of reactants. Okay? You got to explain that, right? I said what we were watching for already. The thing we're watching for is always our responding variable. Okay? We're looking for some things, in particular, after we mix these things together. That's a responding variable. You identify it and explain why. Okay? Controlled variables, 3 of them. Explain each one. Okay? We already said that one of them has to be the amount of everything. Okay? We want to have the same amount. There's a reason for that. You need to figure out what it is. Okay? There needs to be 2 other things that should stay the same throughout the lab. Think about things that might affect how fast a reaction happens, okay? Things like that. Okay? Think about those kind of things as your other 2 controlled variables. Remember to explain each one. For our hypothesis, what 3 words does the hypothesis have to have? Okay, if, and then. Okay, in the if part, you should talk about what a chemical reaction is or what happens in a chemical reaction. A good place to look for that is in Dalton's atomic theory. Because one of his points tells us what happens in a chemical reaction. Something about what can happen to atoms. Okay? It's in one of his points. Okay? That should be what the if part of your hypothesis talks about. If this is what a chemical reaction is, or if this is what happens in a chemical reaction. Okay? The and part is a brief description of the experiment. Okay? So give me a rundown of the procedure. Right? We're mixing things together. Don't say it like that. Okay? But that's essentially what's going on. Talk about that. Then has to tell me what you're going to find and your expected findings and what they're going to mean. Okay? What are we watching for here? Every time I mix things together, I should see changes, right? Certain changes. The signs of a chemical reaction. All right? So you should probably say then that in each pair there will be something about, a chemical change, and maybe list the signs of a chemical change, because that's probably your evidence that one has gone on. Okay. For the initiating and planning, there is a chart that you are going to have to fill out. Okay. That's the watch glass, by the way. That's what they look like. Big giant contact lens. Okay, all the unknowns are going to be on the front table like that. Some of them will be in flasks because they'll be solutions. Others of them will be solids. Okay, and then the waste bucket, that's where everything goes when you're done. Don't put any of this stuff down the sink. Okay, your predictions are something we're probably going to work on right before you go into the lab, and that is you're going to have to predict, based on what we know of chemical reactions, what the products of these reactions are going to be. You're not going to be right on all of them, and that's okay. Okay? In fact, I'm kind of setting it up so that you will be wrong on a couple of them and that you can figure out which ones you were wrong on. Because not all of them go exactly the way we would predict them according to like a double replacement or a single replacement reaction. They don't always happen exactly that way. All right, so we'll talk about that prediction probably tomorrow. Okay. Pair 4 and pair 5, you don't have to predict because it's not a kind of reaction that we actually go over in Science 10, but it does have some value in terms of seeing certain signs of a chemical reaction, so we'll still be doing them, you just don't have to predict or write the reactions for them. Okay, so that's kind of your initiating and planning part there, okay, that you see up here. Then you don't have to write materials, they're already in your lab report because your template looks a lot like the last time. Whoop, sorry, wrong one. Okay, your template looks a lot like the last time. Okay, it's just you got to fill it all in. And I think I actually filled in these ones here already for you. 4 and 5 are already done. Okay, the materials are there. Okay, the procedure is already there. You don't have to write that. You got observations to write in that you'll get during the lab. And then for the analysis, you're writing out the actual reactions. Formulas and words. Okay, both forms. Balanced properly, types identified. Okay, that's your main job in the analysis and interpretation. There's one other question in the analysis and interpretation, okay, and it has to do with the precipitates. 2 of the reactions are going to produce precipitates, and you need to figure out from your reactions which products were not soluble using your solubility chart. Okay, we'll talk more about that when we do the lab. Okay, conclusion, same as last time. Restate your hypothesis, copy paste it. Okay, and then using your results, explain to me why it's acceptable or rejected. Okay, did you see those signs? Did they show what you thought they would show? Okay, and then you got your sources of error, 2, and ways to fix them. All right, so same as before. That's how your lab report should set up. Okay, there'll be a second podcast when we actually do the lab, and I explain a couple more things in a little bit more detail, but that should get you going. All right, are there any questions there? What should you do with this recording when you're putting your lab together? Yeah, okay? Don't have to watch it, just have it playing in the background so you get all those big juicy hints I dropped just a few minutes ago. Okay? All right.